As we progress with the levels of organization unit, I'm going to go back as a revision lesson here to cells. Now, you may remember from the exam question that was given where we were looking at a particular type of specialized cell in the digestive system that was involved in moving substances from different concentration gradients, and that was a specific type of specialized cell. Now, when we talk about cells, you need to be aware that there are essentially two categories of cells, and let's say the various specialized cells, be they plant cells, animal cells, single-celled organisms, can be classified within these two categories. So I'm going to do a quick brief recap of these categories of cells. So the first one are called eukaryotic cells. So could you just put the subheading eukaryotic cells and just put a 1 by it? So this is the first type or one type of category of cells. Now I'm going to list various types of eukaryotic cells because it has been asked in the past in certain exam questions to identify the eukaryotic cell or give an example of a eukaryotic cell. And students tend to get confused when they see these two words, eukaryotic or prokaryotic, which is the other category. So examples of eukaryotic cells: animal cells, plant cells, you've got the next one, protista. So animal cells, plant cells, protista. Now, these are examples of certain of eukaryotic cells. Now, they all have something in common, or so let's say eukaryotic cells also have certain features that are in common. So, can you, to the side of that list, put a dividing line? So we'll just separate this and we'll put some unique features of eukaryotic cells to the side of it. So can you just put a dividing line that separates it, and can you put unique features to the side? Okay, now, so eukaryotic cells have certain features that distinguish them from prokaryotic cells. One of the main distinguishing features of eukaryotic cells is that the DNA is contained within a nucleus. Now, exam questions will invariably ask you, if you are given an exam question comparing eukaryotic and prokaryotic cells, they tend to look for your understanding that the DNA of eukaryotic cells are contained within a nucleus. It will become clearer when we look at prokaryotic cells as to how the DNA is stored and arranged differently in prokaryotic cells. So the DNA, the way the DNA is stored within eukaryotic cells is within a structure known as the nucleus. Now, prokaryotic and eukaryotic cells have a lot of features that are in common, and I will show you this by diagram later on, but I'm repeating again, the identifying unique characteristic of eukaryotic cells is that the DNA is contained within the nucleus. So prokaryotes are said to be single-celled or unicellular living organisms. And the classic example of the prokaryotes, and which you will need to know for your exams, are bacteria. So can you just put down bacteria as your example for prokaryotes? So these are unicellular, single-celled living organisms. Now, they have some, again, when we compare them to eukaryotes, prokaryotes have certain unique features to their group of cells, to their type of cells. So we'll do the same thing. And there are a few more that I want to put onto this one. So bacteria. The first thing is that genetic material is not contained in a nucleus. Now, it's free within the cytoplasm, so it's not contained in one specific organelle. So genetic material is not contained in a nucleus. Second point about the genetic material. Factors for prokaryotes that you have to be aware of are that the genetic material, as well as not being contained within a nucleus, it is free in the cytoplasm. So, as there is no nucleus, the genetic material therefore is in the cytoplasm. Furthermore, bacterial DNA is in a single loop. So this is important. So the bacterial DNA is in a single loop. So this is the DNA. The DNA of prokaryotes, they're not contained within a nucleus. They're free in the cytoplasm in a single loop. The third point about the DNA or the genetic material found in prokaryotes is that they may contain one or more extra small rings of DNA called plasmids. So, may contain one or more extra small rings of genetic material, of DNA. Okay, and these are called plasmids. Now, this is an important statement because these plasmids are going to be important later on in biology when we talk about genetic engineering and how genetic engineering occurs, because these plasmids play a pivotal role in genetic engineering. But for now, I want you to underline these key words here: rings and plasmid. So when you think about small rings within a prokaryotic cell, you should be thinking about plasmids. Now, the last point, which I want you to put down, is that prokaryotes have cell walls. Right? Now, this is similar to plant cells. However, the distinguishing feature for prokaryotes are that there is no cellulose within the cell wall. So cell wall does not contain cellulose. Now, I have seen exam questions where in the question, the build up to the question, there are statements where they give you a cell, but they specifically tell you within the cell that the cell wall, within the question, that the cell wall does not contain cellulose. However, it is a photosynthetic organism, because not all photosynthesizing cells are plant-based. So you should be aware of that. And that's why I'm giving you this statement here. Prokaryotic cells or prokaryotes have a cell wall, but the cell wall does not contain cellulose. Now, there is one more point I want to put here. It's just I'm running out of space on it. So put it at the bottom here. We'll say that prokaryotes may contain flagella. Okay, so prokaryotes may contain or may have flagella. Now, this is like a tail-like substance that allows the prokaryotic cell to propel itself and allows it to move through a particular medium. Now, what we're going to move on to, I'm going to show you a diagram where you can see the eukaryotic cell and the prokaryotic cell side by side. Now, I want you to pause the video at that point, and I want you to just look at the distinguishing features of a typical eukaryotic cell and a typical prokaryotic cell. There are more labels there on the diagram than you need to know. However, it gives you a good idea of what a prokaryotic cell is and what a eukaryotic cell is. Then we're going to move on to the order of magnitude of prokaryotic cells and eukaryotic cells and how we can place them in relation to other known objects of size which we can actually comprehend. So here you can see a eukaryotic cell side by side with two types of prokaryotic cells. Now I have given you two images for the prokaryotic cell to emphasize a point of the flagella, and the singular being flagellum. So you can see here the top prokaryotic cell here, having one flagellum, and some prokaryotic cells have many flagellum, and that's depicted there. Now I want you to draw your attention to this point here, because in case you are asked to identify a particular type of prokaryotic cell and you're not seeing the flagellum, you may be tempted to label it as a eukaryotic cell. But just be aware, as it's mentioned here, that the slime capsule and the flagellum, it's been cut off here, but the slime capsule and the flagellum, these are not always present in prokaryotic cells. So you will have to look carefully at the diagram that you're given if you're asked to identify a eukaryotic or prokaryotic cell by the features displayed. Just be aware that the slime capsule and the flagellum or flagella are not always present. So then your attention has to be drawn to the nucleus or the genetic material, the lack of a nucleus or plasmids. So that's why when we were noting down the key characteristics of eukaryotic and prokaryotic cells, I drew your attention to the genetic material and how the genetic material is stored within these cells. Okay, so here I want you to be familiar with the various features. So you may want to pause the video at this point and take a good look at the labels, look at the internal cellular organelles, and also look at how the genetic material is being stored within these structures, within these types of cells. Okay, now one final label I want to put onto these diagrams, because we're going to move on to the orders of magnitude and to compare the relative sizes. Prokaryotic cells, so can you just make a note of this? I'm just going to put it on this diagram. They have an order of size that's approximately 1 micrometer. Sorry. 1 micrometer. So the order of size is approximately 1 micrometer. Now, your eukaryotic cells are approximately between 100 micrometers to 10 micrometers. So you've got a clear distinction in terms of size between your prokaryotic cells and your eukaryotic cells. So if I just put here prokaryotic, so these are prokaryotic cells and these are eukaryotic cells. So just be aware of the various sizes, the size comparison of your bacterial cells compared to plants and animal cells. Your bacterial cells are many times or order of magnitude smaller than your animal cells or plant cells. Here you can see a typical eukaryotic cell. Now, you may notice some of the organelles that were shown in the diagram represented here. Now, I want you just to take a look at a few key organelles that you need to be aware of. And this diagram gives you a 3D visualization of how certain cellular organelles appear. And I'm going to ask you to focus here on the blue cylindrical cellular organelles. So let's take one of the blue cylindrical ones here. So it says here mitochondrion. Now, here it says a mitochondrion is known as a powerhouse of the cell and produces energy to fuel the cell's activities. And in your note, I want you just to put down that mitochondrion as being the powerhouse of the cell. Now, this is important because of ATP and it's linked to the respiration of a cell. Okay, so the mitochondrion, known as the powerhouse of the cell, produces energy to fuel the cell's activities. So that was a mitochondria. Now, if we look at the nucleus, okay, it holds the genetic material. Now, we can go further into the nucleus and we can see the genetic material there. Now, what I want you to take from here is that the nucleus is where DNA is stored and it is the control center. It controls cellular activities. Mitochondria provide the energy for cellular activities. The nucleus is the control center of cellular activities. Okay, now if we again go on to this diagram and let's look at the cell membrane. Now, the cell membrane, for now, what you need to be familiar with or what you need to be comfortable with regarding the cell membrane is that it controls entry into and out of the cell. Okay, so the cell membrane controls what goes into the cell and also what comes out of the cell. Okay, so cells also need to, in certain instances, let substances move out, and that will become clearer when we talk about certain types of cells that are involved in the defense of the body. But for now, just at this stage, can you put down on a piece of paper that the cell membrane controls entry into and out of the cell. Now, you may be wondering what this strange stacked layer thing here is. This is the Golgi body. So if you go back to the previous diagram I showed you, the 2D representation, you will see this represented there, and this is the Golgi body. Okay, so it is used to modify and, as it says here, distribute proteins that are made in the cells. Okay, so it modifies and it packages and distributes proteins that are made within the cell. Now, I'm trying to get the cytoplasm. We'll see if we can do that. Okay, the cytoplasm is not coming up, but so cytoplasm, okay, is where chemical reactions take place. So just jot down on a piece of paper, the cytoplasm is where chemical reactions take place. So this was just for your reference, so you could just see a typical eukaryotic cell, typical animal cell, in its 3D representation. Sometimes it can get a bit difficult to visualize what you are seeing on a 2D picture. So this allows you to see the 3D representation of your animal cell or eukaryotic cell. I will put the link to where you can get this particular 3D representation in the description below. And then you can go through it in more detail in your own time and look at the various structures. I won't do all of them here, but I will just show you. You can see here the 3D representation of a typical eukaryotic cell. Here you can see a prokaryotic cell, a bacteria, and you can see with the bacterial cells you can see the flagella, and you can see here it is distinct. There is a distinct difference in terms of its structure when compared to your animal cell or your eukaryotic cell. Now, if we were to go closer into our prokaryotic cell, you can see that the genetic material is not contained within a nucleus, it's free flowing. Now, just be aware of that, okay? So this is not a nucleus. You're not seeing a nucleus here. You are seeing genetic material that is not contained within a nucleus. Now, it says the nucleoid is a region of bacteria that contains the DNA. But be careful at this point, because you must, at your level and for your exam, you need to state that the DNA is not contained within a nucleus for bacteria. You've got your plasmid here. So you can see the plasmid there. The ringed section of DNA, circular section of DNA. And it's important for genetic engineering. And that will be, and you will need to know its importance and its function later on when we look at genetic engineering in a bit more detail. So here you have the flagella, okay, a tail-like structure that allows the bacteria to move. Okay, so this allows the bacteria to propel itself in various mediums. Now, if we were to compare eukaryotic cells to prokaryotic cells and even make a more general comparison to cellular organelles or larger structures such as whole organisms, we can do it on a scale which is known as a logarithmic scale. And what we can see is that previously I had mentioned that the cellular organelles were quite different when we compare eukaryotic and prokaryotic cells. Now, the reason being, we can see straight away, if we look at the chart that is horizontal, where we've got an ostrich egg and a chicken egg, and then we're going further and further down the scale in terms of more and more smaller measurements. When we get to the bacteria, and bacteria are prokaryotes. Now look at the scale or the relative size of a bacteria to the mitochondria. Now if you remember back to the eukaryotic diagram, mitochondria are found inside eukaryotic cells. Now if you look here, just an order of magnitude, if you look to the right of your screen from bacteria, you can see that animal and plant cells contain mitochondria as part of their internal cellular structures. And a bacteria or a single cell living organism like prokaryotes are comparably the same size as mitochondria. That is why you cannot find mitochondria inside prokaryotes. You cannot find them inside bacteria. So this is something again that can help you in an exam type question where you're given a diagram and you're asked to identify an unknown organism, and you're asked to determine if it's a prokaryote or if it's a eukaryote. If it's got a mitochondria, it cannot be a bacterial, or it cannot be bacterium. It cannot be a prokaryotic cell, because prokaryotes are on, let's say, on relative size comparisons, the same size as some of the internal cellular organelles such as mitochondria. So I want you to take a look at these two scales here, and you can see where the light microscope has been identified. Now this is important because the light microscope is the range of humans being able to see via using just a normal microscope such as a light microscope. And you can see from a wasp down to approximately, you can see some prokaryotic cells. And then viruses, DNA and molecules become increasingly difficult to see, and that's why we have electron microscopes further down from there.